Somewhere inside a single strand of DNA belonging to one of the most photographed women on the planet, a set of genetic instructions turned against the body that carried them. Not a virus, not an injury, not anything from the outside world at all. The enemy was written into her own code, waiting for years for the right combination of triggers to switch it on. This is the story of Selena Gomez, but it is also the story of something far stranger and far older than fame. The story of how the same genome that can make a person a global icon can also contain a set of molecular instructions capable of turning the immune system into a weapon aimed at its own host. To understand what happened inside her body, we have to go somewhere most documentaries never take you. Not to a red carpet, not to a recording studio, but to the 6-foot length of DNA coiled inside almost every one of her cells, and to the strange, ancient, and only partially understood disease known as lupus. To really grasp what that means, it helps to remember what DNA actually is. Every cell in the human body carries roughly 3 billion base pairs, 4 chemical letters, adenine, thymine, guanine, and cytosine, arranged in sequences that spell out the entire blueprint of a person, coiled so tightly that if you stretch the DNA from a single cell end to end, it would measure about 6 feet. Multiply that by the tens of trillions of cells in a human body, and the scale becomes almost impossible to comprehend. A library vast enough to stretch across enormous distances, all folded into a space smaller than a grain of sand. It took the International Human Genome Project, completed in the early 2000s after more than a decade of work by thousands of scientists across the globe, to finally read that library from beginning to end, giving researchers, for the first time, a reference map of the human genetic code. Without that map, the kind of large-scale genetic detective work that eventually connected lupus to hundreds of specific risk locations simply would not have been possible. Every gene linked to Gomez's condition, every HLA variant, every interferon pathway signal exists as a coordinate on a map that did not exist a generation ago. For most of the world, the story began in the early 2010s, when a young star at the peak of her career suddenly disappeared from the public eye. Tabloids speculated about exhaustion, about rehab, about scandal. The truth, when it finally emerged, was something far more biological and far more universal in its implications. In 2015, speaking with Billboard, Gomez revealed that she had been diagnosed with lupus and had undergone chemotherapy, explaining that this, and not the rumors swirling around her, was the real reason behind her abrupt break from performing. It was a moment that did something rare in modern celebrity culture. It took a disease most people had never heard of and pulled it almost overnight into the center of public conversation. Researchers would later track internet searches for the question, "What is lupus?" and find 3 unmistakable spikes, each one aligned precisely with a public disclosure Gomez made about her condition. A single person's willingness to speak had measurably changed how many people on Earth were trying to understand their own immune systems. But to understand what lupus actually is, we need to shrink down, past the skin, past the bloodstream, into the microscopic battlefield where the disease actually plays out. The immune system is, in essence, a surveillance network built from trillions of cells, each one trained across a lifetime to recognize a simple rule: attack anything foreign, protect anything that belongs to you. B cells patrol the body producing antibodies, precision, guided molecules built to lock onto invaders like viruses and bacteria. T cells act as both commanders and executioners, directing the immune response and destroying infected cells outright. In a healthy body, this system is almost unbelievably good at telling self from non-self. Lupus is what happens when that ancient, evolved distinction breaks down. In lupus, the immune system begins manufacturing antibodies against the body's own tissue, misreading fragments of a person's own DNA, proteins, and cell membranes as threats to be destroyed. The result is systemic inflammation that can strike almost anywhere: the skin, the joints, the kidneys, the heart, the brain. It is, in a very real sense, friendly fire at the molecular level, and it is written in part into the genome itself. For decades, scientists assumed autoimmune disease was simply bad luck, a kind of biological accident with no discernible pattern. That assumption began to collapse the moment researchers started sequencing the genomes of lupus patients on a large scale. What they found was staggering. Today, more than 230 distinct genetic risk locations scattered across the human genome have been linked to lupus susceptibility, identified through genome-wide association studies that compare the DNA of thousands of patients against thousands of healthy controls, searching for tiny variations that appear more often in people with the disease. And the heritability numbers are just as striking. Twin and family studies suggest that somewhere between 40 and 60% of a person's risk for developing lupus is written into their genes, and that people with a close relative who has lupus face a risk roughly 20 times higher than the general population. This is not a disease that strikes at random. It is a disease with a genetic architecture, one that scientists are only now beginning to fully map. At the center of that architecture sits one of the most fought-over regions of the entire human genome, the major histocompatibility complex, a dense cluster of genes on chromosome 6 that includes the HLA family. These genes are, in a sense, the immune system's identification department. They encode proteins that sit on the surface of cells and present fragments of proteins to patrolling T cells, essentially holding up a piece of evidence and asking, is this you or is this a threat? Certain variants of these genes, carried silently by millions of people, appear to make mistakes more likely. One particular variant, HLA-DRB1-03.01, has emerged from decades of research as one of the single strongest known genetic risk factors for lupus, nearly doubling a carrier's odds of developing the disease. Other HLA combinations have been linked to the production of specific autoantibodies, the very molecules doctors now use to diagnose lupus in a blood test. It is a genuinely unsettling thought, the same genetic machinery that gives humans one of their greatest evolutionary advantages. The ability to recognize an almost infinite variety of pathogens can also, in certain configurations, misfire against its own host. But HLA is only the beginning of the story, and this is where lupus genetics becomes something closer to an ongoing detective investigation than a closed case. Beyond the HLA region, researchers have identified genes controlling B-cell behavior, genes regulating a signaling network called the interferon pathway, and genes like TLR7 sitting on the X chromosome that help cells detect viral genetic material. In a landmark study published in early 2026, researchers uncovered a remarkably common genetic variant that heightens a signaling molecule called IRF7, boosting the production of interferon alpha, an inflammatory signal so central to lupus that scientists sometimes refer to the disease's underlying biology as an interferon storm. The variant appears to have persisted in human populations because, in the right context, it helps the body fight off viral infections more aggressively. It is, in other words, a genetic trait that may have once offered a survival advantage against ancient viral threats, and now in a subset of carriers, tips the immune system into turning against itself. Evolution rarely writes in absolutes. It writes in trade-offs, and lupus may be one of the oldest trade-offs written into the human genome. Picture, for a moment, what that interferon storm actually looks like at the cellular level, because it is genuinely one of the more dramatic scenes the body ever produces, invisible to the naked eye but staggering under a microscope. A cell, mistakenly convinced it is under viral attack because of the genetic wiring it inherited, releases interferon alpha into the surrounding tissue. That signal ripples outward, cell to cell, like an alarm spreading through a building, each new cell that receives it ramping up its own defenses and releasing more alarm signals in turn. In a genuine viral infection, this cascade is exactly what the body needs, a rapid, self-amplifying call to arms that helps contain a pathogen before it can spread. In lupus, that same cascade fires in the absence of any real invader, an entire biological alarm system, responding to a threat that does not exist, and in the process, recruiting B cells and T cells into a sustained assault on the body's own tissue. Multiply that scene across kidneys, joints, skin, and blood vessels, sustained not for hours, but for years, and the sheer physical toll of the disease starts to come into focus. This is not a single dramatic event. It is a slow-motion siege, waged by a person's own immune system against their own organs, scripted years or even decades in advance by genetic variants inherited long before symptoms ever appeared. That interferon connection leads to one of the most fascinating and least understood threads in the entire lupus story, the relationship between our genes and the viruses that have infected humanity for millennia. The Epstein-Barr virus, the same pathogen responsible for infectious mononucleosis and carried by the vast majority of adults on Earth, appears again and again in lupus research. Certain genetic variants, particularly in a gene called CD40, seem to change how the immune system responds to Epstein-Barr infection. And recent research has traced a chain reaction in which virus-infected B cells activate helper T cells, which in turn activate uninfected B cells, driving a systemic autoimmune response strikingly similar to what doctors observe in lupus patients. This is precision medicine's next frontier, the attempt to understand not just which genes raise risk, but how those genes interact with a lifetime of environmental exposures, from viral infections to ultraviolet light to hormonal shifts, to finally tip a genetically primed immune system over the edge into full-blown disease. And this brings us to one of the most consistent and most genetically revealing patterns in all of lupus, the disease's overwhelming preference for women. Roughly 90% of people diagnosed with lupus are female, and the disease most often strikes during the reproductive years, precisely the window in which estrogen and other sex hormones are at their most active. Researchers studying this imbalance have found that sex hormones directly influence the behavior of immune cells, altering how aggressively B cells and T cells respond and how much inflammatory signaling gets triggered. Add to this the biology of the X chromosome itself, which carries a disproportionate number of immune-related genes, including that TLR7 gene capable of amplifying interferon signaling, and a genetic picture begins to emerge that helps explain why a disease rooted in DNA would so consistently single out one sex. Gomez's own case fits this pattern precisely. She was in her early 20s, within the classic window in which lupus most often reveals itself, when her body's immune surveillance system began treating her own tissue as the enemy. Genetics also helps explain something else that quietly runs through Gomez's story and through lupus more broadly. The disease does not strike every population equally. Research consistently shows that lupus is more common and often more severe among Hispanic, Black, Asian, and indigenous populations compared to white populations. Disparity that traces back, at least in part, to differences in the frequency of specific risk variants across ancestral groups. Studies of admixed populations in Mexico, for instance, have found that specific HLA configurations tied to lupus risk appear at different frequencies depending on a person's genetic ancestry, layering the story of population. Genetics directly onto the story of autoimmune disease. Gomez, who has spoken publicly about her Mexican-American heritage, exists within a demographic reality that lupus researchers have been trying to untangle for decades. The disease is not colorblind, because the genome is not uniform across human populations, shaped instead by thousands of years of migration, adaptation, and the genetic bottlenecks and expansions that come with them. None of this genetic complexity stayed abstract for Gomez. It became devastatingly physical. The chemotherapy she disclosed in 2015 was not treatment for cancer, but for lupus itself, using a powerful immune-suppressing drug called cyclophosphamide, a therapy so aggressive doctors typically reserve it for cases where the disease threatens major organs. In her case, it did. Lupus can trigger a complication called lupus nephritis, in which the immune system's misdirected antibodies attack the kidneys. And in 2017, Gomez shared that she had undergone a kidney transplant, with her close friend, actress Francia Raisa, stepping forward as the living donor. Buried inside that act of friendship is another entire chapter of genetic science. Every organ transplant is, at its core, a genetic negotiation, doctors screening for compatible HLA types between donor and recipient, trying to minimize the risk that the recipient's immune system will recognize the new organ as foreign and attack it exactly the way lupus taught Gomez's own immune system to attack her original kidneys. It is a strange and moving irony. The same HLA genes capable of misfiring into autoimmune disease are the genes doctors must carefully match to give a transplant any chance of surviving inside its new body. Gomez's account of that period, delivered years later, offered a rare, unfiltered look at what living with a genetically rooted disease actually feels like from the inside, beyond the clinical language of loci and odds ratios. She described the isolation of hiding a serious illness behind public rumor, and the relief and vulnerability of finally speaking the truth once she made the decision to reveal her diagnosis. She has spoken candidly about how the medications used to control her immune system caused her weight to fluctuate, feeding a wave of public body. Shaming she endured before most people understood the disease behind it, once explaining in her own words that she was being criticized for weight gain caused directly by her lupus treatment. What genetics research quantifies in probabilities and risk loci, patients live as fatigue, joint pain, medication side effects, and an ongoing negotiation between a body's own immune defenses and the drugs designed to hold them back. That negotiation, it turns out, rarely ends cleanly. Lupus is understood by researchers not as a condition to be cured, but as a condition to be managed, cycling through periods of flare when symptoms intensify and remission when they quiet down. By 2024, Gomez shared that her lupus had moved into remission, a genuine milestone, though one that comes with an asterisk written into the biology of the disease itself. Remission is not the same as resolution, and patients typically remain on medication indefinitely to keep a genetically primed immune system from reactivating. More recently, she has spoken about developing arthritis connected to her lupus, a reminder that even in remission, the underlying genetic and immunological wiring of the disease, there's another layer to this story that genetics research has only recently begun to untangle, one that complicates the neat picture of genes as fixed destiny. Epigenetics, the study of how life experience can change, which genes get switched on or off without altering the underlying DNA sequence itself. Chronic stress, infection, sunlight exposure, and even the passage of time can chemically tag genes, sometimes silencing protective genes or activating inflammatory ones. For a disease like lupus, where genetic risk alone does not fully explain who develops symptoms and who does not, epigenetics offers a compelling piece of the puzzle. suggesting that a person's environment does not simply trigger a static genetic program, but can actively reshape how that program runs. It is a humbling idea. The genome a person is born with is not a finished script, but something closer to a living document, annotated and re-annotated across a lifetime by everything that happens to the body carrying it. That same principle, genes interacting dynamically with environment and treatment, is exactly what is now driving the next generation of lupus research, and it borrows a set of tools that would have sounded like science fiction only a decade ago. Gene editing technology, most famously CRISPR, allows scientists to make precise, targeted changes to DNA sequences, snipping out or correcting specific genetic instructions, rather than simply suppressing whatever those instructions produce. Early-stage laboratory research is now exploring whether CRISPR-based tools could one day be used to dial down the activity of hyperactive immune genes like TLR7, or to engineer a patient's own immune cells outside the body, correcting their genetic programming before returning them, in a process conceptually related to the CAR T-cell therapies already transforming cancer treatment. None of this is available to patients yet, and any responsible account of the science has to be clear about that distinction. This is emerging, experimental research, not an established treatment. But the trajectory is unmistakable, moving steadily from broad immune suppression toward targeted biologics, toward a genuinely genomic future in which a disease like lupus might one day be corrected at its molecular root rather than managed indefinitely from the outside. Here is where the science becomes genuinely hopeful, and where the future of Gomez's disease, and diseases like it, begins to look radically different from its past. For most of medical history, autoimmune disease was treated with blunt instruments, broad immune suppression, steroids, chemotherapy drugs never designed for this purpose, tools that dampen the entire immune system indiscriminately and carry serious long-term side effects, from bone loss to increased infection risk to elevated cancer risk. The genomic revolution is beginning to replace that blunt approach with something far more precise. Researchers analyzing the genetic architecture of lupus have identified specific genes, among them TNFRSF17 and TNFRSF13B, that encode receptors sitting on the surface of B cells, receptors that respond to signaling molecules called BAFF and APRIL, because scientists now understand this pathway. At the genetic level, pharmaceutical researchers have been able to design biologic drugs that target it directly, selectively quieting the exact immune cells driving lupus, rather than suppressing the entire system. It is the difference between carpet bombing a city to stop a single rogue unit and sending in a team that already knows exactly which building to enter. This is the quiet, unglamorous frontier of genomic medicine, the place where decades of painstaking genome-wide association studies, thousands of research papers, and careful cataloging of risk, loci finally start converting into treatments that reach real patients. Scientists like Judith James and John Harley, whose research groups have spent years mapping the genetic terrain of lupus, helped build the foundation that made this kind of targeted therapy conceivable in the first place. And the frontier keeps moving. Researchers are now exploring how variants in interferon pathway genes might predict which patients will respond best to specific biologic drugs, edging toward a future in which a person's genome, sequenced early, could tell doctors not just that they carry elevated lupus risk, but which treatment pathway is most likely to work for their particular genetic configuration, long before a devastating flare forces the issue. This is also where the story of lupus intersects with one of the most consequential shifts happening across all of modern medicine, the move toward genuinely personalized treatment. For most of the 20th century, medicine largely operated on averages, prescribing the same steroid, the same immune-suppressing drug, to every lupus patient regardless of which specific genetic pathway was driving their disease. That approach is beginning to change, as researchers build increasingly detailed maps of which genetic variants correspond to which biological pathway, whether a patient's disease is being driven primarily by interferon signaling, by B-cell hyperactivity, or by some other combination entirely. The possibility opens up of matching patients to the treatment most likely to work for their particular genetic profile, rather than cycling through medications by trial and error while organ damage accumulates in the background. Some researchers are even exploring polygenic risk scores, statistical models that combine information from hundreds of genetic variants at once to estimate a person's overall lupus risk long before any symptoms appear, potentially allowing at-risk individuals, including close relatives of existing patients, to be monitored more closely from an early age. None of this is standard clinical practice yet, but the direction of travel across the field is unmistakable. Genetics moving from a diagnostic curiosity to a genuine steering wheel for treatment decisions. There is, of course, a sobering asterisk on all of this progress, one that any responsible telling of the science has to include. Despite everything researchers have learned about the genetics of lupus, the disease remains at a Its core, unpredictable. Carrying even a high-risk variant like HLA-DRB1-3-1 does not guarantee a person will ever develop lupus. Genetics loads the gun, in the words researchers often use for complex diseases like this one, but it is the combination of environment, infection history, hormones, and sheer chance that so often pulls the trigger. This is precisely why current diagnosis can still take years, with damage sometimes accumulating in silence before doctors piece together the full picture, and why scientists are so determined to develop earlier genetic and molecular screening tools capable of catching the disease before it reaches the kind of crisis Gomez experienced with her kidneys. What makes Gomez's story resonate as science communication rather than simple celebrity biography is what she chose to do with her diagnosis once she had absorbed it. Rather than retreating from public view, she used her platform, at one point directing followers toward lupus research organizations after sharing news of her transplant, turning a private genetic misfortune into a public engine for awareness and, indirectly, for research funding. Lupus, despite affecting an estimated 5 million people worldwide, has historically received a fraction of the research investment aimed at diseases with comparable impact, and public health researchers have specifically credited high-profile disclosures like hers with measurably are not simply the sum of our choices, our habits, our environments. Written into the 3 billion letters of our genome are silent probabilities, ancient trade-offs between infection defense and self-tolerance, variants inherited from ancestors who survived plagues we can barely imagine, passed down through thousands of generations until they arrived entirely without warning in the cells of a teenager who would go on to become one of the most recognized faces on Earth. Her DNA did not choose her fame. It did, in a very real biological sense, help right the disease that would nearly take her life, and it will, through the ongoing efforts of the researchers decoding conditions, just like hers, help write the treatments that keep patients like her alive and thriving for decades to come. That is the deeper story lupus genetics is quietly telling us, gene by gene, risk locus by risk locus. The immune system that protects us from every virus, every infection, every invisible threat we encounter across a lifetime is built from instructions so ancient and so finely tuned that even a tiny miscalibration, inherited from parents who never knew they carried it, can turn that same system against its own body. And yet the same science, capable of explaining that failure is now, gene by painstaking gene, learning how to correct it, offering patients a future their grandparents' generation could never have imagined. Not a cure, not yet, but a level of precision and a level of hope, written directly into the code that once seemed to be working against them.